lost her first tooth, and that was her school picture.
That's what we call it, snaggle tooth.
Well, because it was funny, the first picture she took, she hadn't lost her tooth yet, and we ended up having to do retakes, so then she lost her tooth in between, so she has like the missing tooth in her school pictures.
Alright, so we start here. I want to show you some optical illusions. So this is the after image effect or negative image that you're asked to do for the eye lab. There's colored pieces of paper over on the counter, or you can do it at home by just cutting out a red piece of paper or a blue piece of paper and putting it on a white background. Staring at it, it says to close your eyes. To do close your eyes, tilt your head back a little bit, look on the back of your retina, or you can just look away at another white space of paper and look into a light. What happens is the photoreceptors become bleached in essence, overused, and when you look away, the ones, the brain becomes adapted to that, and the ones that were only slightly affected by the light that you were looking at is what is reflected off of the white surface and what the eye starts processing. There are a lot of optical illusions out there. So this particular one, it's not part of the lab, it's one that I found that's kind of interesting. So the exercise asks you to count the number of black dots. And I'm not sure if it works. So if you look at one,
They change.
yeah, so they're really all white, but your eye fills it in.
Yeah.
So you see the black dot starting to
Mine are
move
black,
around.
mine are like maroon.
So this is your eye, it's kind of your eye filling in. Alright, so what do you see on the left side image?
Face.
A guy playing a saxophone.
Okay, so there's a face and then there's the musician. What about the upper right hand image?
Lift.
Okay, so you're seeing the contrast.
I didn't even see that at first, I just saw the street. That's creepy.
Young lady. See a young lady or an old hat?
For a little bit.
I see the young lady.
So do I, but then there's a little, like a, well,
Yeah,
Oh,
so
there's
there's
the old
a,
hat.
so there's a young lady with the cat,
yeah, it's an old
and
lady.
then I always have a hard time finding the old lady and then I
Yeah,
can't
so
see
do
it.
I.
So this becomes
Her
the
nose.
nose. So here's the nose and chin,
Yep, see it.
and then the eyes, kind of a bump there for the nose.
I see it.
Yeah.
And the lips.
Okay,
Yeah, that's the eye, yeah.
So the ear becomes the eye of the older lady, of the old woman. Okay? So those are just some of the fun things that you can kind of trick the eye into doing. So let's look at the external ear now, and middle ear and inner ear. Two of these are air-filled spaces and function to amplify sound waves. So the external ear is formed of a pinna.
She put an earring in her ear.
You guys remember another name for the pinna? It was those two bony surfaces that we had between the sacrum and the ilium that looked like ears. Remember what those were called? The auricular surfaces. So this is also known as the auricle, with an AU instead of an O. It has elastic cartilage, and that's what we can bend it forward without breaking it. Now I made this kind of wide because the rest of my ear structures are quite large and rather shortened, but this would be the external auditory canal, and it channels the sound waves rather, towards the middle ear. So the auricle's purpose is to capture sound waves. All right? Before hearing aids were developed, you would see images of people with trouble hearing with a great big metal trumpet that tapered into a narrow tube that they would place in their ear, and that was for the purpose of capturing the sound waves and funneling it in towards the external ear. So people, beauty-wise, like ears best that lay flat against the head. They'll even have surgery to remove some of the cartilage so their ears don't stick out like this. The ones that do are actually better functional in capturing sound. All right, because if they're flat against your head, they're not going to do much for that.
Is it also known as external acoustic meatus?
Yes, acoustic and auditory are the same.
Okay.
Meatus, yeah, so we use that term more often for the bony opening, but either one is fine.
Okay.
In the skin, outside of this, we have glands that secrete wax. into the ear canal. And what's our other word for wax? Does anybody know? Cerumen. All right. So these are ceruminous glands. And I was unaware until just, I think last year when I don't know why I was looking at wax. I thought everybody had the same kind of earwax. I do, which is a kind of brownish, orangey, liquidy stuff. But Asians have a dry, flaky type of wax. All right, so it's not soft and gooey at all. Got one of the morning students said that her husband, who's Japanese, when they first met, he had this bamboo stick with a curve that he was cleaning his ear with. She said, "What are you doing?" He says, "This is how I grew up cleaning my ear instead of Q-tips." So it may be because we have as Caucasians, those of us that are Caucasians have that more moist kind of earwax. It's going to soak up into a cotton, but if you have a flaky one, using a Q-tip isn't going to work very well trying to get a flaky earwax out. And so scraping it might work a bit better. Yes?
One of the issues that can occur with hearing loss, actually a lot of times it's wax, and the soft wax will harden. And the best way to get it is actually to soak with hydrogen peroxide first.
I had a student at Sac State that she said she and her whole family once a year would go to the doctor to get their earwax removed. And she was Asian, so I'm not sure if it was because of that, but she said, and sometimes it feels kind of good to scratch the inside of your ear. There's ear candles that are really, I don't know, I would put a burning piece of paper near my ear, but you can buy them in stores. It's a coil, like a newspaper with a wax, and you light it and it melts the wax that's hardened, so it flows out. So yeah, the Q-tips actually are not supposed to put anything smaller than your elbow in your ear. So Q-tips are really supposed to just clean the auricle, and the problem with putting them into the ear canal is it tends to drive the wax in deeper and pack it.
I'm comparing what you have on the right-hand side to the left-hand
Uh-huh.
side. And
So
I'm
what
kind of
I've drawn
confused.
over here, I just drew I foreshortened my external auditory canal a bit. So this is the auricle.
Right.
This would be the bone of the skull, and this is the external auditory canal, or meatus, and the tympanic membrane,
And
which I haven't quite gotten to.
What's the ceruminous?
The ceruminous glands are not, I don't know if they're even shown here.
They're not.
They're going to be in the skin of
In
the
the
lining
skin.
of the ear canal,
Okay.
and they secrete into the ear canal.
Okay, thank you.
Okay? So then the very last structure of our external auditory ear is actually the eardrum. And another name for the ear, that's kind of the layman's term, like your shoulder blade or your breastbone versus the name of the bone. And so the name of this structure is actually the primary tympanic membrane. This one over here is the secondary tympanic membrane. This is our first conversion of the energy of sound waves. So sound travels in waves, okay, in frequency. And this is the first conversion of those sound waves to a mechanical wave. And so the sound is like you hit a drum, okay, and it vibrates. The tympanic membrane is going to vibrate, and this results in an amplification of that energy. You'll notice it's tinted inward towards the middle ear cavity. In some individuals, it is flat, it's more flat shape, or it may be inverted outward in this direction. And that flatness or reverse tinting actually decreases the air pressure in the middle ear cavity and can function to draw fluid from your throat up into the middle ear cavity and increase the incidences of middle ear infections or otitis media. So this is the eustachian tube or internal auditory tube. And it opens up in the throat directly posterior to your nasal cavity. There are muscles that attach between your palate and the lower opening of the auditory tube in your nasal cavity. So yawning or stretching or swallowing can help to open up that space. Okay. Because of the connection between the pharynx and the middle ear cavity, parents are usually or should be taught not to give their babies bottles when they go to bed. Of course, if they have teeth and juice, that can help to ruin the teeth. But if they don't even have teeth yet and they're old enough to hold a bottle, drinking in that supine position can increase the amount of that milk and mucus that ends up in their middle ear. All right. Now, one of my nephews, when he wasn't quite 2, developed a severe ear infection. His parents had to wait about a week or so before they could fly back from visiting my parents. And so when they did get back to their home, they took him in, and that's when they found that he had one of those inverted, shaped eardrums, and so he had several ear tubes. They're small but not microscopic. My youngest son, remember, was born with his jaw that was too short, and also the innervation of the muscles when he swallowed were affected. So although his hearing was normal when he was born, 2 months later, when they tested him and they bounced sound off of his membrane and measured how much it vibrated, and he had lost 50% of his hearing. Not because he had lost any nerves or structure, but his auditory tube wouldn't open. And since when that was closed, the air in the middle ear cavity was reabsorbed and replaced by fluid. Even though the fluid was sterile and it wasn't an infected fluid, it created a resistance to the vibration of the primary tympanic membrane and decreased his sensitivity to sound. So at the age of 7 months, they put in 3 or 4 tubes, and I found one in his crib or his bed when he was about 3. So they just kind of fell out, and his nerve and muscle function developed and overcame that.
I was going to ask, couldn't they drain that fluid?
Yeah, but it would just so by placing the tubes, that allowed the airways to move, the air to move into and prevent the fluid from replacing it.
Where was the fluid being produced?
The fluid was being produced by the lining. This is an epithelial lining. So it was just passing through, seeping through the epithelial lining into the space. This is a serous fluid. All right, so that's the external ear. And the tympanic membrane, the primary tympanic membrane is a component of that. Then we move into the middle ear, which is also an air space. It's filled with air. And so it's this space that you see right here. So we have the reverse side of the tympanic membrane forming one wall of it, the bony structure of our inner ear. Here's actually the opening to the mastoid sinuses. Remember we talked about the mastoid sinuses? That's how an ear infection can get from the middle ear through to the mastoid sinuses. And we have 3 ear ossicles. There's 2 of these, so if you have one near you, start passing it around. These are actual ossicles, bones of the ear. They're often described as the smallest bones, and I'm going to show you a video where the stapes is called the smallest bone in the body. It's not actually accurate because we have 2 sesamoid bones on the bottom of our big toe that guide the tendon, the muscle that we use to flex our toe. That's smaller than the stapes, about half the size of the stapes. But pass that around. So these ossicles are further going to amplify the vibration. So the first one is actually attached to the surface of the eardrum, the medial surface of the eardrum, and you can see it here, attached to that medial surface. All right, this is actually a different view. So let me draw those on here. And this is known as the malleus, which means hammer. Okay. Attached to the malleus is a similarly shaped structure that is known as the incus, which means anvil. So the hammer is hitting on the anvil. And then finally, let me change the shape of this a little bit. The incus is attached to that stirrup-shaped one. And our word for stirrup is stapes. Well, not my word, but I'm assuming it's Latin. So these are the 3 ossicles of the middle ear. So when the tympanic membrane vibrates, it causes the malleus to move, that causes the incus to move, and that causes the stapes to move. So all of that, again, is amplifying the vibration created by the sound waves. However, sometimes we don't want this. We want to be able to control or suppress the degree of the vibration. And so there are a couple of muscles within the middle ear. This is kind of looking from this view. There's one that goes to the malleus, and it comes up in just above the middle ear, just above the internal auditory tube. So this is a slightly different view, but this is known as the tensor tympani. So it's going to dampen vibration of the tympanic membrane, innervated by trigeminal nerve. And the other one dampens the movement of the stapes, and it's called the stapedius. So not difficult to differentiate between these. This is skeletal muscle, not smooth muscle, but the movement is done by reflex. We don't decide to tighten or loosen our tensor tympani and stapedius. All right, so here's this is the internal auditory canal here, and so coming in just above that, tensor tympani and stapedius. Oval window membrane on the diagram next is right there. So this is all middle ear. So a middle ear infection is an infection, buildup of pus and fluid inside the middle ear. Treating that with ear drops doesn't work. Okay, you can treat an external ear infection with ear drops, something you get swimming or something like that. But if it's within the middle ear, then you have to take systemic antibiotics. And it's very controversial whether to give children antibiotics or anybody antibiotics for ear infections. That they go away about the same rate whether you give them or you don't. There are some children that seem to have them all the time, and especially if they're very young, starting say 6 months of age to up to 3, that can affect their ability to speak because it affects their ability to hear if they're not being able to move the eardrum or move the bones properly. So that's the major concern in clearing up the ear infections. So now all the rest of this structure that we have here belongs to the inner ear. And this is where we actually have receptors that are activated for hearing or that are activated for balance. So remember the name of the cranial nerve that's involved with balance and hearing? Vestibulocochlear. All right? So I kind of blunted the curvature of this so I could diagram it a little bit more better, but this is known as the cochlea. This is the utricle and saccule here, and I'm going to draw these separately when we're done. And then the semicircular canals. So the cochlea is for hearing, and the utricle and saccule, the utricle, saccule, and semicircular canals are for balance. So we're going to focus on hearing first. So these structures, have you ever heard of earworms? You know what an earworm is? No. It's a sound, a song that you can't ever get out of your head. You just hear it over and over and over. Okay. But if you consider a real worm that was to be inside your temporal bone, I know it's gross, and kind of bore a hole through it, it would create a bony cavity in the shape of the tan structures that you see here. Kind of like the rubber tube of a bicycle tire. And then inside that rubber tube, we place an inner tube that we pump up with air. So within the bony channel is a membranous tube, and both of them are filled with fluid. So a cross-section of this coiled tube that is the cochlea, or it could be the coiled tube that's a semicircular canal, is shown over here. On the outside of that, we would have a fluid called perilymph, meaning around. And on the inside, within the tube structure, we have the fluid called endolymph. And they have differing concentrations of ions. All right? So if I draw this outer bony channel, and then an inner membranous channel, that's the pattern that we see with the inner ear. So the bony channel contains perilymph, and the membranous channel contains endolymph. Now I'm going to slow down a little bit because we're going to talk about the functional structure of how we hear, and then I'm going to show you a couple of videos I think that explain it three-dimensionally and might be a little bit easier to understand. So sound waves come in through past the auricle, are focused by, not really focused, but channeled by the external auditory canal and cause the primary tympanic membrane to vibrate. As that moves back and forth, it causes the malleus to move, which causes the incus to move, which causes the stapes to move. All right, the stapes is resting on a membrane that is the same shape as the stapes, and so it's difficult to visualize. This is known as the oval window. Right. And it's filled with perilymph. That perilymph is connected through this opening up here to perilymph at the bottom area, so it kind of can form a flow in this passage. So as the stapes is vibrating, it sets up a vibration in this fluid. Okay, so this is going to be perilymph. This is perilymph, and this is endolymph. We're going to get a little bit more detailed here if this seems rather vague. I just want to explain this structure before I move a little smaller. So as the stapes is moving against this fluid, all right, it's creating a fluid wave against the oval fluid. That's going to create a fluid wave that sets up that energy flow, and wherever that wave, as it moves through here, is going to result in the activation, creation of action potentials, but that wave energy needs to dissipate. And so the purpose of this structure here, the secondary tympanic membrane, also known as the round window, is to allow that fluid energy wave to dissipate into the middle ear cavity. So we set it up here with the vibration of the stapes, and then the energy passing through the cochlea can dissipate into the middle ear. Now, I'm going to pause here for a second while you guys go and get some ear models to look at, because this three-dimensional kind of structure is a little bit easier if you look at the ear models. And I'm going to turn off the overhead and diagram the cochlea a little bit larger and in cross-section, and then we'll see all of this in a couple of videos.
Oh no. I didn't put that in the right spot.
Same professor?
Uh-huh.
What is that word?
Helicotrema?
The black one, right there.
Balance.
Oh, that's what's for.
So uterocotactile and semicervical canals function in balance.
Look at that.
I stole the big one. I don't know where it's
Right here.
So cochlea means shell. And we have this long process here
Welcome.
that I drew over there, kind of coiled around about makes 2.5 coils.
Okay, I need some chocolate right now.
So I'm redrawing the cochlea kind of uncoiled. We have have the oval window and the round window. On your ear models, you will not be able to see the oval window very well because the stapes is going to be sitting on it. All right, but see if you can find where the stapes is attached to the oval window.
You have the oval and round one.
So here's the stapes, and underneath that would be the oval window. And if you look just nearby will be the round window. Okay?
So oval window is under staples. The oval window's on top, the round is downstairs.
Yeah. Uh-huh.
Okay, so oval.
All right? So remember that circle I drew with the bony tube and the membranous tube inside it? So I'm going to draw that here in blue this time to match the colors that I drew before. So when we vibrate this, we're vibrating fluid in this space right here. This space is known as the scala vestibuli. The space down here is known as the scala tympani. So the oval window is closing off the scala vestibuli, and the round window is closing off the scala tympani. Okay. The round window is actually not an open window, it's a membrane. The secondary tympanic membrane, and this would be the oval window. Okay. This space here is the scala media. So this would be where the endolymph is, and this would be perilymph around it. So all three spaces are going to be filled with fluid.
So say where the fluid is again.
Perilymph, endolymph, and perilymph again. So I'm writing the word scala media within the scala vestibuli actually, but it's within the two blue lines. Again, moving that diagram from over there to here. So this would be the scala media, right? And scala vestibuli and scala tympani. Now this coils around a central core and actually creates, if we were to cut through this, so we cut through it in cross section, it creates an image that looks like this. It's really not a true circle in the middle. But we would have the, so this would be the scala media. Okay. Actually, this comes down a little bit more like this. And then we have tissue here, bone and structure. So that's the scala vestibuli. This is the scala tympani. And this would be the scala media. also known as the cochlear duct. So I generally refer to it as the scala media. So these two membranes are then given a name. So this membrane is known as the vestibular membrane, and I'll transfer this to the other diagram to help. That's the vestibular membrane. And this would be the vestibular membrane.
Timpani membrane?
That's vestibular.
No, the bottom
This is
one.
actually the basilar.
What?
I know.
Just had to throw the wild card in there.
So that's the basilar membrane. Okay. And then we have another membrane known as the known as the tectorial membrane. It only comes out partially into the scala media. So if I were to draw it here, however, on this longitudinal view, now resting on the basilar membrane are a series of cells that are tall cells. Some of them are supportive cells, some of them are receptors for sound. And out here, they have, some of them have tall folds of plasma membrane. They're really long microvilli, which we know as stereocilia. These are known as the outer hair cells. They're not really hair, they're long microvilli. And these are the inner hair cells. As you'll see in the video, the outer hair cells actually move to help further increase the vibration that's going to be caused by the sound waves. Whereas the inner hair cells have neurons. These are going to form the cochlear portion of our vestibulocochlear nerve. So these are carrying sound waves, action potentials created by the sound wave converted to mechanical wave to converted to fluid wave that we hear as sound.
Just going back to the tectorial membrane, is that just serving as like kind of like a mediary between the ciliary fibers on each side? Like how, trying to figure out how this is connected.
Okay.
Like when it gets to the end, like the arc of the horn. Is it connected at the end over there?
The
or?
basilar membrane, yes, it's connected. The basilar membrane, the tectorial membrane can move like this. You'll see it in a moment in the video.
Okay.
So what happens, let me put in just kind of a little bit of the hair cells here. So when we hear a sound, causing our external auditory membrane to vibrate, stapes vibrates after all the bones vibrate. That sets up a fluid wave in the perilymph of the scala vestibuli. If that fluid wave is very high in frequency, so a high-pitched noise has a higher frequency, all right, it's going to cause a vibration of the basilar membrane close to this opening. So we're going to see this. We're going to see a fluid wave that does this. And it's going to push on that basilar membrane, and it's going to push these cells up into the tectorial membrane, causing those long microvilli to be bent. That opens channels, ion channels, and we see a depolarization of the cell, resulting in an action potential, if it's strong enough, that gets carried into the vestibulocochlear nerve and to the temporal lobe. So the purpose of the basilar membrane is to support the neurons and the inner cells. The purpose of the tectorial membrane is the mechanical bending of those microvilli. Now, so high frequency sounds are heard close to the base. Low frequency sounds have a lower pitch, and they are going to travel up a little bit farther before their force actually pushes up on the tectorial membrane. So we hear low sounds out here, high pitched sounds down here. One of the soccer moms that I used to visit with at my son's practice told me about how the night before she had been cleaning her ear with Johnson & Johnson Q-tips. Those are the ones that are hollow plastic, not the cheap cardboard ones. And it must not have been wrapped properly, and they're hollow. The plastic went right through her primary tympanic membrane and into her round window. So she didn't pull it out, she just left it there. I said, did you bump your elbow or anything? She said, no, it just cut right through. And so went to the doctor, and there was some bleeding and stuff, but they pulled it out and gave her antibiotics. I said, I bet you've got some hearing loss of high-pitched noises, because that's where we process that information, is right next to the round window. And she said, yes, but that's okay because I was too sensitive to high-pitched noises anyway. So let's look at a couple of videos. Why don't you go ahead and take your pictures while I get this set up. The red is just showing the fluid waves. Oh, that's the afferent neurons carrying the sensory information to the vestibulocochlear nerve.
I have an idea in my
Those would be the
head of
first
like
order
what
neurons of
the
various
membrane does. It's almost like bouncing. I know you're good. You know what's, that's a small. That hurts. I probably did that much, but you go get a chicken nugget out of there.
Are they from an adult or a child?
Actually, they're probably from adult, as you hear in the video. They don't change size.
Oh,
They stay the same size.
You like your eyes on the cigarettes?
Yeah.
It's interesting. A lot of names, but it's
It's magnetic, the way that it sticks to my fingers, charging down magnetic.
Megan knows. And you were sitting right there too. You were probably out of it too. It's cool. That's right. On the other side. Don't worry about it. Yeah.
I bet you go.
That is something.
It's Cobra. Is that empty now? You're just gonna leave that right there, huh?
I mean, not because it's
I know lots of people who says that. It's like,
Like none of my don't fit on the truck.
Until they, you know, yeah, it's probably more likely the reason why they hurt is.
Because I'm just not used to it. There's no way you're going to put it good enough that it's going to fit on.
Okay, let's take a look at this
Like,
video.
I didn't measure it.
I have a second one after that. Actually, two more, but there's one that's much more long.
Transmitted to the organ of Corti and translated into neural impulses to be delivered to the brain. The organ of Corti is located in the cochlea, a spiral, three-chambered, snail-like structure, 10 millimeters wide, within a bony matrix.
So the yellow would be the nerve.
The organ of Corti extends from the anterior part of the vestibule and coils for about 2.5 turns around a bony pillar called the modilus. In cross-section, the uppermost chamber is called the scala vestibuli. The oval window is situated at the base of this chamber. The lowermost chamber is called the scala tympani. At the base of this chamber is where the round window is located. Both the scala vestibuli and scala tympani contain perilymph. Between the scala vestibuli and scala tympani is the scala media. This houses the organ of Corti, which is referred to as the receptor organ of hearing. The scala media is filled with endolymph. The scala media includes structures from the tectorial membrane, basilar membrane, and hair cells, which sense the mechanical forces. The hair cells are located between the tectorial and basilar membranes. Approximately 16,000 hair cells are within the cochlea. There are two kinds of hair cells, inner and outer hair cells. 95% of the afferent fibers are from the inner hair cells, the sensory receptors that communicate with neurons from cranial nerve 8. Outer hair cells receive mostly efferent input from the superior olivary complex. The filamentous structures that connect the tips of adjacent stereocilia are known as tip links. These are thought to amplify the forces in the area of the molecular sensors. How does sound enter the cochlea? Compression hits the tympanic membrane, causing the stapes to transfer force to the oval window. The sound travels down the scala vestibuli, around the helicotrema, to the scala tympani, allowing its fluid perilymph to mix. From there, sound moves to the round window. High frequencies are encoded at the base, and low frequencies at the apex. It is this property that leads to the tonotopic map along the basilar membrane. The manner in which the basilar membrane vibrates in response to sound is the key to understanding cochlear function. The hair cells are located between the tectorial and basilar membranes and are stimulated by the shearing force between the two caused by the pivot point of the two membranes. The pivot point of the basilar membrane becomes displaced. The tectorial membrane moves across the tops of the hair cells, causing the stereocilia to bend. The ionic environment of the compartments plays a critical role in signal transduction.
This goes on and on for a while.
The apical portion of the hair cell is bathed in high potassium solution, and the base of the hair cell is bathed in potassium poor solution. This causes the opening of mechanosensitive channels, allowing potassium to flow into the cell, leading to depolarization.
No, it's potassium that's depolarizing the cell, not the sodium that we're typically used to, or even calcium.
This in turn opens calcium channels at the basal end of the cell, leading to vesicular transmitter release to stimulate the nerve. Because the relative voltage and potassium levels are low at the base of the hair cell, potassium flows out of the cell. This establishes that potassium flow through the cell is used for both depolarization, potassium in at the apex, and repolarization, potassium out at the base of the hair cell.
look at those events.
How I'm hearing you right now, Joe, is crazy.
Collects the sounds that surround us and channels them down the air canal, vibrating.
You hear this okay?
On the other side of the drum, these vibrations are transmitted through the little air by minute bones.
Uh-oh.
These are the only...
I know.
An adult has air bones the same size as a newborn baby. One of them is the stirrup. It's about the size of a grain of rice. The stirrup rests on the oval window of the inner air and passes vibrations through it. On the other side is something rather intriguing. Called the cochlea, it's a small, bony, spiral tube. It's the key to how we hear the vast range of sounds that we do. If we zoom right the way in with an electron microscope,
It's like driving in a garage.
remarkable. The secret of how sound is transmitted to the brain. They're rows of minute hairs, only a few thousandths
They're not
of
really
millimeters
hairs.
high,